Yes, sir.
Can
Yes,
we,
sir.
yes.
Yes.
Can we move on to the next system? That is the essentials or introduction to the gut or GIT.
Yes, sir.
Thank you. So let us open a new chapter. I will only go to the essentials of GIT because of limited time. So I would go which functions of the GIT, that is gut secretion, digestion, and absorption of food substances, right? So if the time permits, we may have glimpses of the gut hormones as well. So look here, this is the oral cavity. So the question is, which digestive events occur in the oral cavity, in the mouth? The mouth, we all know, secretes saliva. And what is saliva? Saliva is the secretion of parotid glands, submandibular glands, or sublingual glands, as you can see in this diagram. So sublingual glands, submandibular glands, and parotid glands secrete, and their secretion is called as saliva. Saliva is, now what does saliva consist of? Which cells of the parotid, sublingual, and submandibular glands? So they are the acinar cells. Acinar cells of the sublingual, submandibular, and parotid glands secrete saliva. What does saliva consist of? It consists of a rich enzyme called as salivary amylase, or called as ptyalin, P-T-Y-L-A-I-N. Ptyalin, or salivary amylase, is the main enzyme in the saliva. And it is rich in water, rich in electrolytes. It is rich in sodium, rich in bicarbonate, rich in chloride, but poor in potassium. So it contains mainly water, electrolytes like sodium, bicarbonate, and chloride, and consists of salivary amylase or ptyalin. So now this is the reflex pathway of salivation. When we say it is a reflex action, so it should have all the components of a reflex arc, starting from stimulus up till response. So what is the stimulus for secretion of saliva? So that is presence of food or other object in the mouth. Sometimes even thinking of food, of a favorite food will lead to salivation. Even looking at the cooking of some food may lead to salivation. And even for barbecue, even to smelling the smell of the barbecue, of the smoke, right, typical, would lead to salivation. Even the thinking of the time of meal, that is 1 at noon or 2 at noon, will lead to salivation. So that means it is presence of food or objective plus certain psychoneurological features, certain psychoneurological stimuli as well. And these stimulus are received by the transducer, which are the receptors, which are located in the oral cavity. From there, afferent neurons, that means sensory neurons, that is the trigeminal nerve, which is 5th cranial nerve, to the facial nerve, which is 7th cranial nerve, glossopharyngeal nerve, which is 9th cranial nerve, and the vagus nerve, which is the 10th cranial nerve. So these receptors receive the stimulus and convert it into signal, message, information, or an impulse. And this impulse is taken by the sensory afferent neurons to the center, that is to the synapse. Where is the salivary center located? It goes to the salivary centers in the medulla oblongata of the brain. From there, motor fibers or efferent nerve fibers, which is craniosacral nerves, that is parasympathetic, within the 7th and 9th cranial nerve. So it is a parasympathetic response within the 7th and 9th cranial nerve, and these efferent or motor nerve fibers, they go to the effector tissues, which are the salivary glands, which we have just enumerated to stimulate them to cause secretion of saliva. And then this is an enzyme containing lubricant is poured into the mouth, which is the main enzyme, that is salivary amylase. So which digestive activity would occur in the stomach? Gastric digestion. Like in mucosa of in the mucosa of the stomach, there is rugae. The stomach is accommodated in a very small space, like in the form of rugae. So they provide a large surface area for stretching of the stomach, and it allows for the distension of the stomach. As we keep on drinking and keep on taking meals, so the stomach wall gets distension because rugi, they start opening up. And it consists of certain important cells. They are mucus cells, they are peptic cells, and they are oxyntic cells. Mucus cells secrete mucus, and the main function of the mucus is the emollient action. So that is, it gives a coating on the gastric mucosa to prevent digestion or erosion of the gastric own wall from its own HCl and from the acidic meals. And it secretes intrinsic factor of Kasal, and this intrinsic factor of Kasal is required for absorption of vitamin B12. If total gastrectomy is done, it leads to vitamin B12 deficiency, and there are features of pernicious anemia. And then it is peptic cells, like it secretes peptic cells. Peptic cells secrete like a substance called as pepsinogen. Pepsinogen, like pepsinogen, its active form pepsin, like so pepsinogen is related with protein digestion. And it secretes gastric lipase, like which gastric lipase enzyme, which can only digest triglycerides containing short chain fatty acids. Like, and then there are oxyntic cells which secrete HCl. Now this HCl affects on pepsinogen to convert it into pepsin, and pepsin is responsible for digestion of protein food substances. Digestion and absorption occurring in the small intestine. Now, a small intestine, like small intestine, lumen of the small intestine, does not secrete any hormone or any enzyme of its own, but mucosal epithelial cells, they secrete disaccharidases. Like, so disaccharidases enzyme are produced by the epithelial cells of the small intestine. But small intestine now gets bile from the main and accessory, so gets bile from the liver, like it gets bile from the liver, and it gets pancreatic juice through the main and accessory pancreatic ducts. So lumen of the small intestine now receives pancreatic juice, receives bile, and the epithelial cells of the small intestine, they secrete enzymes, disaccharidases. So pancreatic juice, as we all know, is rich in enzyme. It is alkaline, it is rich in bicarbonate, and it is rich in enzyme. So there are two peculiarities of the pancreatic juice. It is strongly alkaline because it is rich in bicarbonate, and it is rich in enzymes. Which enzymes? Trypsin, chymotrypsin, and carboxypeptidase. So these three enzymes are responsible for digestion of protein food. So trypsin, chymotrypsin, and carboxypeptidase, enzymes of the pancreatic juice within the lumen of the small intestine, like digests protein food substances. And then pancreatic juice has also got pancreatic amylase. So when amylase is there, it is concerned with digestion of carbohydrate. And then pancreatic juice, main important pancreatic juice enzyme is pancreatic lipase. The main beauty of the pancreatic juice is of pancreatic lipase. And this pancreatic lipase now is responsible for digestion of fat particles, which we are going to study in a while. Like, so that means pancreatic lipase for digestion of fat food substances, pancreatic amylase, digestion of carbohydrate, trypsin, chymotrypsin, and carboxypeptidase are responsible for digestion of protein. And then is the liver bile, like bile salts. Which are the bile salts? Sodium glycolate, sodium tarocolate, potassium glycolate, potassium tarocolate, or the bile salts. So these bile salts are responsible for digestion of fat. First of all, bile salts will act upon the fat particle to have priming effect, and then pancreatic lipase can exert its action. And then by the epithelial cells of the small intestine, disaccharidases, enzymes are produced, and these disaccharidases converts disaccharides into monosaccharides. It converts disaccharides into glucose and galactose. So with these words, now which are the summary and the digestion activities of the large intestine. When I say large intestine, I essentially mean colon, not rectum, but colon. And please mind, it is the sigmoid colon where the fecal material gets stored up. Now it is the stomach where the food gets stored up. So they have got these two, colon, sigmoid part of the colon, and this stomach have got elasticity, plasticity properties. Like they stretch without increasing appropriate intragastric or intracolic pressure. Like, look at here. Now the large intestinal cells, they secrete mucus. They lubricate the colon to provide like protection against the hard texture of the fecal material. So they lubricate the colon to protect the mucosa from the harmful effects by the solidification of the food particles, digested food particles, into the fecal material. And then as it absorbs water, like please mind 65% of water is absorbed from the small intestine. That is the duodenum and jejunum and ileum. Like it is only 35% that gets absorbed, not 25% from the large intestine, but it's by this absorption of water, the fecal material becomes solidified. And there are certain bacteria in the colon, good bacteria, which have got very much useful effect. They produce certain vitamins, for example, vitamin K, and they produce some bacteria like which converts stercobilinogen into urobilinogen, and minerals are obtained. So the toxic substance, and then the toxic substances are sent to the liver for detoxification. So it is a harbor of the bacterial action, harbors of the bacteria. Undigested carbohydrates, proteins, and amino acids are broken down so that they can be expelled in the feces or absorbed and detoxified by the liver. Certain vitamins, like vitamin K, is synthesized from the good bacteria in the colon. Now, this is a slide which is a crowded slide, but is a very useful slide, and it's a beautiful slide. So I will go slow because some of you may not be able to read it. Now, this slide is the summary of all the actions of the enzymes, from where they are secreted, how much quantity is secreted, what is their optimal pH for the action, and which action is catalyzed by them in terms of digestion. So see the upper column of the slide. Now, upper column of the slide on your left-hand side is the enzyme called as salivary amylase. So that means this is for the oral cavity. Salivary amylase or TALN. It is secreted by the salivary glands. Which are the salivary glands? Sublingual, submandibular, and parotid gland. How much quantity is secreted? 1200 ml per day, or roughly you can remember 1 liters per day salivary gland secrete. And what is the normal pH for secretion of saliva? 0.66 to 8. That means from highly acidic to highly alkaline, because 7.4 is the normal pH of the plasma in humans. It varies from species to species, from acidic to alkaline, 6 to 8. And salivary amylase is inactivated by gastric HCl. So what does it do? With digestive activity is performed by salivary amylase. It converts cooked starches. Atta ko nahi, roti ko yes. Cooked starches, it converts cooked starches into maltose. Maltose is a disaccharide. So it converts cooked starches into maltose. If it is an uncooked starch, so that will not be digested in the oral cavity, right? And then look at the second from above, pepsin. Pepsin is a chemical which is secreted by the stomach. How much? 2 liters per day. So salivary secretion, 1 liter per day. Pepsin from the gastric juice is 2 liters per day. And look at its pH. pH, it requires pH of 1 to 2, highly acidic. And this acidity is provided by the gastric HCl. So gastric HCl affects on the pepsinogen and converts it into pepsin. And this pepsin affects on the proteins and breaks down them into polypeptides. Look at the third block of the slide from above. That is regarding the pancreatic juice. How much pancreatic juice is secreted in a day? that is again 1 liter per day. Like which enzymes? Trypsin and chymotrypsin. Like trypsin and chymotrypsin. So they require a pH of 7 to 9, which means from neutral to highly alkalinity, neutral to alkalinity. What does it do? They convert trypsin and chymotrypsin, convert chymotrypsinogen to chymotrypsin. So that means they are responsible for digestion of proteins. And then look at the lipase enzyme, pancreatic lipase. pH required is 7 to 9, neutral to highly alkaline. But pancreatic lipase will affect only when the fat molecules are primed up or have been acted already with the bile salts. Like what will they do? They will convert fats into fatty acids and glycerol. And then is pancreatic amylase. So that pH is 6 to 7, from acidic to neutral. And what will it do? It will convert starch into maltose. And then look at the duodenal mucosal epithelial cells. Duodenal mucosal epithelial cells, they produce various, now how much it is secreted? 3 liters per day. So saliva, 1 liter per day, gastric juice, 2 liters per day, pancreatic juice, 1 liter per day, and duodenal mucosa, 3 liters per day. It secretes an enzyme enterokinase. pH optimal is 6 to 9, from acidic to highly alkaline. It converts trypsinogen to trypsin. And duodenal mucosal epithelial cells, they secrete disaccharidases enzyme. So disaccharidase, maltase will affect on maltose to convert it into 2 molecules of glucose. Lactase disaccharide will affect on lactose to convert into glucose and galactose. And whereas sucrase will affect on sucrose to convert it into glucose and fructose. And lipase will affect on fats to convert them into free fatty acids and glycerol. And then peptidases enzyme, they convert polypeptide into dipeptides and dipeptides into amino acids. So this is the summary total of all the enzymes within the oral cavity, within the stomach, within the lumen of the small intestine, receiving bile and pancreatic juice, and in the duodenal mucosal epithelial cells, which are responsible for digestion of protein, fat, and carbohydrate food substances. Now are the very important made easy slides. Like if you can draw this slide afterwards, after this lecture, you will never forget, and you don't have to read a book for your MCQs for carbohydrate digestion. So this slide, look at carefully and try to digest it, is a summary of carbohydrate digestion. If you can understand and draw it by yourself without looking at the slide, then you will never be able to forget digestion of carbohydrate. Look at the upper column of the slide. Upper column of the slide is oral cavity. So that means digestion of carbohydrate starts from the oral cavity. Look at the middle block of the slide. Middle block is of the lumen of the small intestine, and it is receiving some secretion from the pancreas. So that means there is no stomach in between the oral cavity and lumen. So this gives one more information that no active digestion of carbohydrates occurs in the stomach, right? And then look at the lower column of the slide, mucosal cell membrane. So that means digestion of carbohydrate occurs in the mouth, in the lumen of the small intestine, and in mucosal cell membrane. It does not occur actively in the stomach. So in which form we take, in which form carbohydrates are present in the food particles? They are present as monosaccharides, which is glucose and galactose. If you take white, like grinded sugar, white crystal grinded sugar, so that is glucose or galactose, so that does not need any digestion, it will be absorbed into the portal circulation. That is why it is more harmful, because it will immediately switch blood glucose level without any digestion, immediately gets absorbed. but it is a wonder drug against hypoglycemia. If there's hypoglycemic, give him grinded white sugar, crystallized sugar, and the blood sugar will start rising up within minutes to hours. And then there are disaccharides like lactose, sucrose, as milk contain lactose. So lactose and sucrose, they are not acted in the oral cavity. They are not digested, as you can see the arrow on your left-hand side. They are not digested in lumen of the small intestine. So lactose and sucrose, disaccharides, they go into the mucosal cell membrane. And then in the oral cavity, if we take starch, like we take roti or bread, double roti or roti, which contains starch. If we take starch, so if it is a complicated starch, it will not be digested in the oral cavity. That will straight away pass on to the lumen of the small intestine. If it is a cooked starch, roti jaise hai, theek hai ji, not double roti, but roti. If it is a cooked starch, it will be affected by salivary alpha amylase or salivary Talon enzyme, and these starches will be converted into disaccharides. Disaccharides are maltose, maltotriose, and dextrine. So these maltose and maltotriose and dextrins. And then if it is starch, like complicated starch or uncooked bread, so then it will be passed on to the lumen of the small intestine. In the lumen of the small intestine, because it is receiving pancreatic juice, and pancreatic juice has got also an enzyme called as pancreatic alpha amylase. So this pancreatic alpha amylase enzyme will affect on the starch, will convert them into disaccharides. Which disaccharides? Maltose, maltotriose, and dextrin. So these disaccharides, they will pass on to the mucosal cell membrane. Mucosal cell membrane, maltotriose will be recycled, and maltose and dextrinase and dextrin will go to the mucosal cell membrane. In the mucosal cell membrane, they will be affected by secretion of the mucosal cell epithelium of disaccharidases, which are maltase and dextrinase. Maltase will affect on the maltose and will convert it into 2 molecules of glucose. And dextrinase will convert dextrin into 2 molecules of glucose. Glucose is a monosaccharide, and it will be absorbed into the portal blood or portal circulation. Look at the disaccharides, lactose or sucrose, which has been passed from the oral cavity to the lumen, and from the lumen, it has been passed to the mucosal cell membrane. Mucosal cell epithelial cells, they are rich in disaccharide enzyme like lactase and sucrase. So lactase enzyme will affect on the lactose to convert it into glucose and galactose. So milk is acted by lactase enzyme to convert it into glucose and galactose, which are absorbed in the portal circulation. And sucrose will be affected by disaccharide enzyme in the mucosal cell membrane to be converted into glucose and fructose in turn will also be converted to glucose. So which are the monosaccharides? Glucose, fructose, and galactose are the monosaccharides, and they will be absorbed into the portal circulation. So this is the summary of digestion of carbohydrate. Look at this slide silently for 30 seconds and try to absorb it. And then on the similar lines, I will move on to summary of digestion of proteins and summary of digestion of fat molecules. Look at it for 30 minutes in a state of meditation, this slide, so that you can absorb and draw it at end of the class. Please. Right. Can you all hear me, please?
Yes, sir.
Yes, sir. Okay, let's move on the similar lines. Now, again, you don't require much of biochemistry, right, for your MCQs for your part 1 examination. If you can grasp this slide, understand it well, and you can draw it yourself without looking at the slide afterwards, so it will never be forgotten from your mind, and that is what is required in your MCQs. Of course, there's no end to knowledge. The details, you can study them afterwards if you're working in a gastroenterology unit. Look at it, the upper block of the slide is of stomach, not of oral cavity. Which information do we get from the slide right away? So that means there's no active digestion of protein which occurs in the mouth, which occurs in the oral cavity. So that means saliva does not cause active digestion of proteins, right? The middle block is of small intestinal lumen, receiving pancreatic juice. So that means active digestion occurs in the stomach, in the mucosal lumen of the small intestine because of the pancreatic juice, and in the mucosal lower block is of the mucosal cells of the small intestine. So active digestion of the protein particles occurs in the stomach and lumen of the small intestine because of the pancreatic juice, and then into the mucosal cells. No active digestion occurs in the oral cavity. So proteins, if there are complicated proteins we take, they are not absorbed by the stomach. Complicated proteins, they pass on from the stomach, as shown in the left column of the slide. They pass on from the stomach to lumen of the small intestine. But if there are non-complicated proteins in the stomach, they are affected by pepsin. Now, pepsin, as I've told you, pepsinogen is acted by gastric HCl to be converted into pepsin. So pepsin will affect on the proteins to convert them into polypeptides and amino acids. So from the stomach, proteins and polypeptides, they pass on to lumen of the small intestine. Lumen of the small intestine is receiving pancreatic juice, from the main and accessory pancreatic duct, and this pancreatic juice is rich in enzyme trypsin, chymotrypsin, carboxypeptidase, and elastase. So trypsin, chymotrypsin, carboxypeptidase, and elastase enzyme affects on the proteins to convert them into peptides, and it affects on the polypeptides to convert them into amino acids. The amino acids on the extreme left column of the slide, amino acids are absorbed as such in the portal circulation. Amino acids can be absorbed in the portal circulation, whereas these peptides, they pass on to mucosal cell, and these peptides are converted to peptides. And these peptides in the mucosal cell, epithelial cells, secrete an enzyme called as peptidases. So these peptidases enzyme affect on the peptides and convert them into amino acids. So that means protein, part of the food particles, they are digested in the stomach and lumen of the small intestine due to pancreatic secretion, and in the mucosal cell, and they're converted to amino acids. So protein is absorbed into the portal circulation in the form of amino acids. And no active digestion of protein particles occur in the oral cavity. Look at this slide, very carefully in meditation for 30 seconds so that it gets imprinted on your neurons. Can we move on to the next slide, please? Can you all hear me, please?
Yes, sir.
Yes, sir.
Yes, sir.
Yes. Now, this is a difficult slide. This is a difficult slide and requires a lot of attention. And I will go slow. Because lipid digestion is the most complicated. And in MCQs or in questions, like students usually make a mistake. Summary of digestion of lipid particles. Look at the two blocks in this slide. One is of lumen of the small intestine. The other one is the mucosal cells. So there is no block of the oral cavity and of the stomach. So what that is, we get a very useful information. No active digestion of lipid, no active digestion of lipids occur in the oral cavity or in the stomach. Active digestion of lipid occurs in the small intestine. Where? In the lumen, because it receives liver bile and because it receives pancreatic juice. And then it occurs also in the mucosal cell because mucosal epithelial cells secrete certain substances and enzymes. Right? So let us see what events of digestive events occur in the lumen of the small intestine. Lumen of the small intestine does not secrete any enzyme of its own. Its beauty is it is receiving bile from the bile duct and it is receiving pancreatic juice from the pancreatic ducts. Like now fat is present as a triglyceride, a triacylglycerol. So triglycerides are acted by the liver bile, which components of the liver bile salts, which are the bile salts, sodium taurocholate, potassium taurocholate, sodium glycolate, and potassium glycolate. Like, so triglycerides are acted by the ingredients of the bile coming from the liver, the bile salts, lecithin, and cholesterol. So bile salts, lecithin, and cholesterol attacks or acts on the triglyceride in the lumen of the small intestine. This agitates triglycerides molecule. As a result of this, an emulsified form is formed called as emulsion, causes emulsion. Like, so this emulsion is acted by pancreatic lipase enzyme. So pancreatic juice is rich in pancreatic lipase enzyme. So this emulsified material, so pancreatic lipase cannot affect on its substrate if it has not been agitated under influence of bile salts superadded with lecithin and cholesterol. So pancreatic lipase will affect on the emulsified triglycerides to break them into monoglycerides and free fatty acids. So monoglycerides and free fatty acids will be acted by fat-soluble vitamins, vitamin A, D, E, and K. So that means if there's deficiency of vitamin A, D, and K, it will disturb the digestion of lipid particles. So then foul, large, smelly, fecal material will come out, which denotes the undigested lipid food particles. Like, and then these monoglycerides and free fatty acids again are acted by the liver bile. Which component of the liver bile? Again, they are attacked by the cholesterol and the bile salts. And they are changed into micelles. So what are the ingredients? What does a micelles? Within various micelles, like formed up, it is formed in the lumen of the small intestine. What are the ingredients of the micelles? It is monoglycerides, free fatty acids, fat-soluble vitamins, cholesterol, and the bile salts. And these micelles, then they are transported from lumen of the small intestine into the mucosal cell. On its journey, on its transport way, the bile salts, they get recycled. The remaining particle goes to the mucosal cell. So what is left behind in the mucosal cell is again monoglycerides, free fatty acids like fat-soluble vitamins, cholesterol, and bile salts has been recycled. Like, and then in the mucosal cell, from its cholesterol, so cholesterol like fat-soluble vitamins and lecithin will come out. And this particle is re-esterified in the form of triglycerides. So monoglycerides, free fatty acids in the mucosal cell of the small intestine after cholesterol, fat-soluble vitamins, and lecithin has left them, they are re-esterified into triglycerides. And then these triglycerides are again acted by the cholesterol, fat-soluble vitamins, and lecithin, and this structure is ensheathed in a protein covering, and it is a macromolecule, big molecule, macromolecule, and is called as a chylomicron. Chylomicron is singular and chylomicra is plural. What are the ingredients of a chylomicra? Right? Chylomicra has got triglycerides, cholesterol, fat-soluble vitamins, lecithin ensheathed in a protein covering. Right? So that means much of the lipid particles, they're absorbed in the form of chylomicra. If they are short-chain free fatty acids, they can be absorbed into the portal circulation, but abundant bulk of the fat or lipid food particle is absorbed in the form of chylomicra. Since chylomicra is a macromolecule, it cannot be absorbed into the tiny capillaries of the portal circulation. It is absorbed into gutters of the body, and gutters of the body are the lymphatic channels or the lymphatic duct. So chylomicra is the absorbed form of the lipid digested food particles in the lymphatic channels or in the lymphatic vessels. Now look at this slide for 1 minute in a state of meditation, like so that it gets imprinted on your mind.
श्री chylomicrons वाला part एक दफा repeat कर दें.
Chylomicron, no. Micelles from the lumen of the small intestine, they are transported to the mucosal cell. On its transport, bile salts are recycled, and then the breakdown of micelles because of bile salts coming out is changed into and then in the mucosal cell, cholesterol, fat-soluble vitamins, and lecithins, they also leave it. So left behind is monoglycerides and free fatty acids. Monoglycerides and free fatty acids, they get re-esterified into triglycerides, and then triglyceride is acted by, again, cholesterol, fat-soluble vitamins, and lecithin, and it is ensheathed in a protein layer called as chylomicron. Chylomicron is a macromolecule. What are the ingredients of chylomicron? It has got triglycerides, cholesterol, fat-soluble vitamins, lecithin ensheathed in a protein covering. And chylomicron is the absorbed form of the lipid particles in the lymphatic channels, in the lymphatic vessels. Right. So let us move on to absorption of the food particles. So this was digestion. We have known so far that carbohydrates, they are absorbed as a form of monoglycerides in the form of glucose and galactose. We have studied the protein particles, they are absorbed in the form of amino acids. We have studied that most of the lipid particles, they are absorbed in the form of chylomicron. So let us see absorption. This slide shows absorption of the food particles. On your left hand side column is lumen of the small intestine. In the middle column with many villi and microvilli of the small intestine, and then is the epithelial cells of the villus of the small intestine, and your right hand side is the tiny blood capillaries or the portal circulation or the lacteal of a villus. Lacteal of a villus in the small intestine denotes the lymphatic vessel or the lymphatic channel. Look at the upper column of the slide on your left hand side. It is written as glucose and galactose. Glucose and galactose, they are actively transported from the lumen into the mucosal cell, and by the process of passive transport or diffusion, they get absorbed into the tiny capillaries of a villus of the portal circulation. Fructose is a monosaccharide. By the process of facilitated diffusion, it goes from lumen to the epithelial cells of the villus, and then by diffusion or passive transport, it is absorbed into the blood capillary. Look at the middle column, amino acids. Amino acids is the digested form in which it gets absorbed the protein particles. Amino acids are actively transported from lumen to epithelial cells, and by passive transport or diffusion, they are diffused into the capillary of the villus in the portal circulation. Dipeptides, and tripeptides, go from the lumen by the process of active transport into the epithelial cells. They are converted to amino acids, and amino acids are diffused, or by the process of passive transport, they enter into the tiny capillary of a villus. And then short chain fatty acids, by the process of diffusion, they go from the lumen to the epithelium, and then from the epithelial cells of the villus, again by the process of diffusion, they are diffused into the capillaries of the portal circulation. Now the long chain fatty acids and monoglycerides from lumen of the small intestine, they form the macellus, and these macellus are diffused into the epithelial cells of the villus. Here, they're re-esterified into triglycerides, and then this triglyceride is ensheathed in a protein covering. It makes a macromolecule, so chylomicron is formed in the epithelial cells of the villus, and this chylomicron, a macromolecule, is absorbed to the lacteal of the villus, which means into the lymphatic vessels of the small intestine. So this is how food particles are absorbed from the stomach into the general circulation. Now, how they are transported in the general circulation. What is their fate in the general circulation? Look at it like the upper cycle, that is the amino acids, monosaccharides, which is glucose and galactose, and short chain free fatty acids. Amino acids, short chain free fatty acids, glucose and galactose, they're absorbed into the tiny capillaries of the portal circulation, and into the heart, they join the venous circulation. Whereas you can see the lacteal of a duct, and in one of the villus of the small intestine, this lacteal of the duct is nothing but a lymphatic vessel, lymph channel, lymph vessel. So chylomicron are the macromolecules. They are absorbed into the lymphatic vessels, into the lacteal of the villus of the small intestine, and eventually it joins the venous circulation, like now in the inferior and superior vena cava. Right? So this is how the foods are carbohydrate, fats, and proteins are digested and biochemically absorbed and circulated in the plasma to be recycled through the venous circulation. Right? With these, we have got some time. So let us have some glimpses of hormones of the GIT, hormones of the gut. Many of the gut peptide hormones are the hormones of the gut. They are peptide hormones, like endocrine cells. But many of the hormones of the gut, they are neurotransmitters as well, because they are secreted by the central nervous system peripheral nerve endings. They are secreted from the gut endocrine cells, and they're secreted from the peripheral nerves of the central nervous system. For example, substance P, somatostatin, vasoactive intestinal peptide, neurotensin, cholecystokinin, and enkephalins. So they are hormones, they are also neurotransmitters. So that is why they are called as gut-brain peptides. So they are peptides, they are hormones as well as neurotransmitters. When they're secreted from the nerve endings of the nervous system, they are called as neurotransmitters. But when they're secreted from the gut endocrine cells, they are called as hormones, gut hormones. Let us see. Now we are not concerned with the structure. This is the outline of the gastrin hormone, gastrin. They are all peptide hormones. Nobody in part 1 is going to ask you about their structure. Only remember one thing, They're peptide hormones, so they act mainly through cyclic AMP system. So where is gastrin formed? Gastrin is formed in the antrum along the duodenum in human. It is secreted from the antrum in response to what stimuli? In response to protein meal or vagal stimulation. Like vagal stimulation, parasympathetic stimulation, or protein meal stimulates the antrum to cause secretion of a protein hormone of the gut called as gastrin. What does it do? Gastrin stimulates gastric acid secretion, gastric HCl. It increases antral motility. It is a trophic influence on the stomach and intestine. So gastrinoma, or increased gastrin secretion, is one of the major causes of diarrhea in Zollinger-Ellison syndrome, right? And then is cholecystokinin pancreozyme. Again, protein, peptide hormones, from where they are secreted? From the duodenum and upper jejunum. Which are the stimuli for their secretion? That is selective amino acids, long-chain fatty acids, and intraduodenal calcium. You can ignore this. Selective amino acids and long-chain fatty acid stimulate the duodenum and upper jejunum cells to cause secretion of CCK and PZ. What does it do? It causes contraction of the gallbladder to evacuate bile. It stimulates the pancreatic juice secretion, and it makes them rich in bicarbonate and enzyme. Stimulation of pancreatic enzyme to be rich in pancreatic lipase and to be rich in bicarbonate to make it alkaline. And then there's a secretin. This is secretin, like this is secretin. So secretin, secretin is secreted from the duodenum and upper jejunum, and it is a duodenal acidic perfusion. If there's more gastric HCl and there's more evacuation of gastric HCl into the duodenum, so this will be a stimulus for the cells of duodenum and jejunum to cause secretion of secretin. Now, it stimulates pancreatic juice to be rich in watery and rich in bicarbonate. This is not H2CO3, this is in fact HCO3. Rich in alkalinity by bicarbonate and rich in water. It augments the effect of cholecystokinin on pyloric contraction and pancreatic secretion. Secretin, gastrin stimulates, whereas secretin inhibits gastric acid secretion. And it stimulates small intestinal secretion. There's gastrin, gastric inhibitory polypeptide, GIP. GIP is secreted from the whole of the small intestine in response to the mixed meal. What does GIP do? It inhibits gastric acid HCl secretion, inhibits motility of the stomach, inhibits motor activity of the stomach. It is an insulinotropic in the presence of hyperglycemia. Insulinotropic means it stimulates secretion of insulin from beta cells of the pancreas in response to hyperglycemia. It inhibits water absorption in the small intestine. Gastric inhibitory polypeptide. And then there is VIP, vasoactive intestinal peptide. This hormone is secreted from the whole of the small and large intestine, and it is also a neurotransmitter because it is secreted from the cells of the central nervous system and the peripheral nerves. Response is neural stimulation. So there is a new specialty which is coming, is neurogastroenterology. Neurogastroenterology. So some of the conditions, some of the problem in the stomach, they're coming from the brain, they're coming from the neurotransmitters. Neurogastroenterology is a new specialty. So that is from the neural stimulation. And it stimulates intestinal secretion of fluids and electrolytes. It inhibits gastric HCl secretion. It is one of the major mediator in certain watery diarrhea syndromes. And then is motilin. Motilin is a peptide hormone secreted from duodenum and jejunum in response to fall in pH. If there is more acidity coming from gastric HCl by gastric evacuation into the duodenum and jejunum, it stimulates motilin. Motilin stimulates gastric motility and hastens gastric emptying. So with this, we are very much in time, rather before time, like a few words about principal actions of the liver. Like a formation and secretion of bile. You know the beauty of the bile. So bile is bile salts, which are responsible for agitating the triglycerides to form the emulsified material. Bile salt, sodium glycolate, sodium potassium glycolate, sodium tarocolate, and potassium tarocolate help in digestion of the fat particles. It's nutrient and vitamin metabolism. And how does it do it? By glucose and other sugars are formed. Amino acids are formed in the liver. Lipids like fatty acids, cholesterol, lipoproteins are formed in the liver. And then there is fat-soluble vitamins, vitamin A, D, E, and K, they are formed. Even vitamin-soluble vitamins, water-soluble vitamins, like they are also formed, like vitamin C, for example, vitamin B and vitamin B12, like they are also formed in the liver. And then inactivation of the various substances. Inactivation, it is detoxification. Toxic material in the gastric cells is getting detoxicated. Now, steroid hormone, they are inactivated. As we have studied, that cortisol and aldosterone, they are converted to 17-hydroxy ketosteroid and excreted in the urine. So steroids are rendered to their inactive metabolite for excretion in the urine, in the feces, in the saliva, in the sweat, in the seminal flow, in the menstrual flow, in the tears, like, and they are excreted, but mainly from the urine. And other hormones, they are inactivated in the liver. Main site of inactivation of all the hormones is the liver, and main channel of excretion is the urine, that is by the kidney. Synthesis of plasma proteins, acute phase proteins, plasma proteins. All the plasma proteins are formed in the liver except the gamma globulin, immunoglobulins, acute phase. Like all the clotting factors, all the clotting factors are proteins except the calcium. Like, so all the plasma proteins, like, are all the clotting proteins are clotting factors, they formed in the liver except the calcium. And then albumin is mainly formed in the liver. And you know the properties of albumin. Like it tries to regain water within the capillary. If there's hypoalbuminemia, water will leak out, there will be increased ECF volume, and there will be cellular dehydration. Clotting factors, but less calcium. All the clotting factors or clotting proteins are formed in the liver, less calcium. And then steroid binding and other hormone binding proteins, as we have seen, like the hormones, they circulate in the plasma in bound form to globulin, albumin, and pre-albumin. So these are formed in the liver. It is only gamma globulin which are not formed in the liver, which are the immunoglobulins. And then liver produce the immunity as well. From where? By the Kupffer cells of the liver. Kupffer cells of the liver form the antibodies. Now, principal functions of the gallbladder. Gallbladder does not form bile on its own. Like bile is formed by the liver, but it is stored. Storage of bile occurs in the gallbladder. Concentration of bile to 10 times occurs in the gallbladder. Like, because liver bile is so much an amount, so that comes to the liver, ingredients remain the same, water leaks out, so it is 10 times concentrated gallbladder bile. And then under the effect of cholecystokinin enzyme, like there is contraction of the sphincter of the Oddi with evacuation of the bile. And gallbladder also secrete mucus to provide emollient action to prevent it from the abrasive effect of the bile salts. So storage of bile, concentration of bile, evacuation of bile, and secretion of mucus are the major functions of the gallbladder. Look at here. Sometimes the MCQ is also set as liver bile resembles with the gallbladder bile and that. Or the phrase of the question could be gallbladder bile resembles with the liver bile and that. Now, we are not concerned to remember their actual values of the concentration. We have to know which component out of all these ingredients is more or less in the gallbladder bile as compared to the liver bile. So look at water. Water is reabsorbed or absorbed from the gallbladder. So water content are less in the liver bile, in the gallbladder bile as compared to the liver bile. Bile salts are much more concentrated in gallbladder bile than in the liver bile. Bilirubin is much more concentrated in gallbladder bile as compared to the liver bile. Cholesterol is more in concentration in gallbladder bile as compared to that in the liver bile. Fatty acids are more concentrated in the gallbladder bile as compared to the liver bile. Lecithin is more concentrated in the gallbladder bile as compared to the liver bile. Look at sodium. Sodium is reduced. Sodium is low in gallbladder bile as is in the liver bile. Potassium is more high in gallbladder bile as compared to liver bile. Calcium is much more in gallbladder bile as compared to that in the liver bile. Chloride is low in gallbladder bile as compared to the liver bile. Bicarbonate are low in gallbladder bile as compared to liver bile. The summary is water, sodium, chloride, and bicarbonate. Like water, sodium, chloride, and bicarbonate, they are low in gallbladder bile, whereas rest of the ingredients, they are more concentrated in the gallbladder bile. And what is the composition of the liver bile or gallbladder bile? It consists of mainly water, then it consists of bile salts, bilirubin, cholesterol, fatty acids, lecithin, and electrolytes, which means sodium, potassium, calcium, chloride, and bicarbonate. Right. Look at here. Difference between, this is pathophysiology, difference between hemolytic jaundice and obstructive jaundice. Like hemolytic jaundice is, old terminology is prehepatic jaundice. Obstructive jaundice, old terminology is post-hepatic jaundice. Like, so serum bilirubin is high with more free bilirubin in hemolytic or prehepatic jaundice. It is high with more conjugated bilirubin. Bilirubin conjugated with glucuronic acid and with sulfates. So bilirubin conjugated with glucuronides or with sulfates is called as a conjugated bilirubin. And conjugated form of bilirubin is excretable. Like, and urobilinogen in the liver. We have studied today in the large intestine, that is in the colon, the good bacteria converts stercobilinogen into urobilinogen, and then this urobilinogen is passed on to the renal tubule. Like this urobilinogen in the liver, in the urine, is more in hemolytic jaundice. It is absent in obstructive jaundice. So you can differentially diagnose like with this concentration. Urobilinogen in the liver, it is more than normal in hemolytic jaundice. It is absent in post-hepatic or obstructive jaundice. Look at bile salts and bile pigments. Bilirubin and biliverdin, they are the bile pigments. Bilirubin and biliverdin, they are the bile pigments. Sodium glycolate, potassium glycolate, potassium tarocolate, and sodium tarocolate are the bile salts. So bile salts and bile pigments in the urine in hemolytic or prehepatic jaundice, they are absent, but in posthepatic or obstructive jaundice, they are present. Van den Berg reaction, like it is a reaction which depends on different coloration. If it is, no, they only ask for you for interpretation. Like it is, if it is indirectly positive, it denotes hemolytic jaundice. If it is directly positive, if it gives changes of color directly with some chemical, it is directly positive. If it does not give color directly, and then you have to add another ingredient to give the color, that is called as indirectly positive. Like Van den Berg reaction is indirectly positive in hemolytic jaundice, and it is direct positive with post-hepatic or obstructive jaundice. Like if the bile duct are blocked with a stone, that is the post-hepatic or obstructive jaundice. Liver functions in pre-hepatic jaundice, they are normal. Like because liver functions are disturbed in hepatic jaundice, hepatocellular jaundice. Like it is a pre-hepatic jaundice. Like so that means in hemolytic jaundice, they are normal, but in post-hepatic jaundice, they are exaggerated. They are exaggerated, liver functions. And hemorrhagic tendency is absent in pre-hepatic jaundice, present in post-hepatic or obstructive jaundice due to lack of vitamin K. Due to lack of, because the colon bacteria, they synthesize good vitamins, particularly vitamin K. So we are much before time, alhamdulillah, because I increased speed in the early session of today, which was regarding adrenocortical. Now let us overview, and then we will switch on to question answer session. You can make your questions enormously because we have got time. Right? So we started today, what we did, we started with functions, abnormalities, and adrenocortical functions of the adrenal cortex with special emphasis on cortisol and aldosterone. Then we switched on to only the important topics like, I will say contextual introductory topics of the gut, especially of the gut secretion, gut digestion, and gut absorption, and gut absorbed material circulation in the plasma from the oral cavity, in the stomach, in the small intestine, and in the mucosal epithelial cells. And then we move on to hormones and neurotransmitters, which are secreted by the nerve and by the gut. Overviewed them, and we did enumerated only the functions of the liver and functions of the gallbladder. And we did touched slight pathophysiology of differentiating between hemolytic jaundice and obstructive jaundice with the help of certain laboratory tests. So we are very much before time. Any question, please? Yes, please.
Sir, I have a question. Sir, how are exogenous steroids excreted from the body?
Look at here. Because liver functions are normal, so exaggerated liver functions would appear because there is obstruction and the excretory material are not being excreted, right? And then the liver will work more, the liver may secrete more as if the substances are not being excreted, with the view that it gets a false signals that probably the substances are not being digested and absorbed. By the fast feedback, negative feedback system, though the substances are more, but it gets a false signal, the liver cells get a false signals, probably they are not being digested and absorbed, the substances, so they have to get more enzymes to do it. But you can see the book, especially don't go for the contents in the book, you should go for index of the book for the liver functions in post-hepatic jaundice, or go for the post-hepatic or obstructive jaundice and see into the liver functions. You can learn more. But my answer will be certainly incomplete. So go into the index for the post-hepatic or obstructive jaundice and see the subtitle or liver functions in it. You can learn more. Thank you. Any other question, please?
Sir, I have a question. Sir, how are exogenous steroids excreted from the body? I mean, are they excreted through urine or through they are lipid soluble?
देखिए, whether they are exogenous steroids, whether they are produced within the body, they have first of all to be rendered to their metabolites and in the liver, and then they have to be excreted in the urine. And then the metabolites of the steroid hormones mainly are 17-hydroxyketosteroid. So cortisol, adrenaline, and androgens, they are mainly excreted in the form of 17-hydroxyketosteroid in the urine. And this activity occurs within the liver because liver is the main site for metabolic activity of converting them into their metabolites. So whether there is exogenous steroids or endogenously produced steroids, they are inactivated in the liver. Main hepatic cells and by the Kupffer cells, but the main hepatocellular cells by biochemical mechanism. And the main excretory product is 17-hydroxyketosteroid, by which they are excreted mainly in the urine. Other excreta of the body as well, but mainly through the urine. Yes, please. Any other question, please?
सर मुझे एक क्वेश्चन है मेरा कि सर ये कोगुलेशन फैक्टर जो हमने जिक्र किया कि लिवर के उसमें ना वो कम हो जाते हैं, तो ये मुझे सर काइंडली ये हमें बता दीजिए कि प्लेटलेट्स जो हैं वो लिवर डिजीज में क्यों कम हो जाते हैं? थैंक यू।
प्लेटलेट्स लिवर डिजीज में क्यों कम? मेन थिंग इज वेयर आर प्लेटलेट्स फॉर्म्ड। पहली बात तो ये है, वेयर आर प्लेटलेट फॉर्म्ड, पहले तो ये देखेगा, वेयर आर दे फॉर्म्ड?
सर बोन मैरो
एंड
हेमाटोपोएटिक स्टेम सेल्स के थ्रू जो है ना वो फॉर्म।
सर क्लॉटिंग फैक्टर लीवर में बनते हैं, तो प्लेटलेट लीवर में।
जी, तो that is why when the liver cells are damaged, of course there must be certain ingredients of the liver which may be required, like to formulate platelets from the bone marrow cells, So when liver is damaged, so platelets they tend to be decreased. Certain ingredients of the platelets, agar आप platelets की composition देखें, which are the proteins present in the platelets? बाकी ingredients के अलावा, can anyone tell to me?
Platelet-derived growth factors?
Yes. So their proteins are formed in the liver. So when liver cells are damaged, some of the ingredients of the platelets, they are not there. And when they are not there, the platelets counts will also decrease. Formation becomes absent, but it is decreased because certain ingredients of the platelets are formed in the liver.
Excuse me, please. Sorry to interrupt. There are 3 to 4 questions in the chat box. Please, 2 to 3 questions in the chat box.
पढ़ लीजिएगा वरना वो disconnect ना हो जाए मुझसे. I'm not good in IT. वो chat box वाले पढ़ लीजिएगा. I'll give the answer. Try to give the answer. Yes. Just a minute. मैं go chat box. Yes. One question is impair the absorption of vitamin B12 by salts and iron. हाँ जी, इसको मुझे जरा थोड़ा सा explain कर देंगे. What do you mean by this question? Impair the absorption of vitamin B12?
No, sir. Excuse me, sir. The question is like that. Resection of terminal ileum will impair the absorption of distal ileum. Sorry, resection of distal ileum. It will impair the absorption of bile salts or vitamin B12. What should be the answer?
Resection of the terminal
distal
ileum.
ileum. Yes.
Distal ileum, vitamin B12, bile salts, and iron. I will go for iron. What do you think?
So distal ileum में vitamin B12 की absorption होनी
Vitamin
चाहिए?
B12.
लेकिन आयरन भी होता है वहां से बहरहाल. Anyway, bile salts are out. Bile salts are out. So the choice is between vitamin B12 and iron. Now see in the book, because I may not mislead you. It is either vitamin B12 or iron. Both are done, but see which is the main absorption in terms of quantity. Is it of vitamin B12 or iron? See in the book.
Sir,
Yes, sir,
it's
B12
vitamin
is bigger.
B12.
B12.
Major
Okay,
answer
then,
is bigger.
that is fine. Then put your weight in the pan of vitamin B12. अच्छा दूसरा जी
Does it-
absorb जी फरमाइए.
Yes.
हाँ पढ़िए.
So absorption of long chain fatty acids is greatest in ileum, stomach, duodenum, colon, jejunum. Long chain
क्या कहते हैं?
fatty acids.
दोबारा से
Long
पढ़िए.
chain fatty acids greatest absorption.
हाँ
Long
हाँ
chain fatty acids.
ठीक ना. किस में ऑप्शंस क्या हैं?
Ileum, stomach, duodenum, colon, jejunum.
I'll say ileum or
Ileum.
jejunum. The choice is between jejunum and ileum.
Okay.
Choice is
And
between
so
jejunum.
which organ forms peptide which helps in bone growth?
Which organ forms peptides?
Which helps in bone growth?
Organ.
Kidney, liver, brain, intestine.
Oh, especially liver.
Liver?
Liver.
Why not intestine, sir?
no.
Calbindin 128K.
आप पेप्टाइड्स की बात कर रही है ना?
जी. Calbindin भी
Peptides.
peptide.
Which organ
वो
forms?
bone peptide which helps in bone growth.
हाँ नहीं जी,
Liver?
liver. इसमें बोन सेल्स भी हैं?
नहीं सर, bone ऑप्शन नहीं है.
नहीं, लिवर. जब पेप्टाइड फॉर्मेशन की बात आएगी ना, फर्स्ट ऑर्गन शुड स्ट्राइक योर माइंड इज द लिवर. लेकिन उसके बाद देखें अगर कोई प्रोटीन स्पेसिफिक की गई है तो उसकी फिर स्पेसिफिसिटी हो सकती है विद अदर टिश्यूज. अदरवाइज पेप्टाइड जब प्रोटीन की फॉर्मेशन आए तो लिवर. ठीक है.
Okay, so there is another question. A patient had resection of terminal ileum. What will result? Decreased
हाँ, अब
fat
तो आपने बता
absorption.
दिया. अब आपने तो खुद बता दिया पर्निशियस एनीमिया, विटामिन B12. आपने खुद बताया कि टर्मिनल आइलियम.
नहीं सर, इसमें different options हैं.
जी.
Decreased fat absorption, increased water content of feces, increased bile acid concentration in enterohepatic circulation, decreased
ठीक.
absorption of iron, or increased fat absorption.
Increased fat absorption. ये है increased fat absorption आप कह रही हैं?
जी सर मैं पूछ रही हूँ, ये ऑप्शंस ये हैं जी. लास्ट ऑप्शन है increased fat absorption.
Decrease in vitamin B12 है ना ये? स्टेम क्या था?
A patient had resection of terminal ileum, so what will result? Decreased fat absorption, increased water content of feces, increased bile acid concentration in enterohepatic circulation, decreased absorption of iron, or decreased fat or increased fat absorption.
Increased fat absorption.
Increased होगी sir.
इसको देख लीजिएगा जी. At random, I'm not catching it, so I don't want to mislead you.
इसमें एनीमिया का क्या जिक्र है? दोबारा रिपीट करिएगा.
Anemia का जिक्र नहीं है. The question is,
Anemia
a patient had resection of terminal ileum. So what will result?
Decreased
Decreased
absorption
fat.
of iron है ना इसके अंदर?
नहीं, the answer is increased water content of feces.
Okay.
इसको जी देख लीजिएगा बुक में. I don't want to confuse you and don't
सर
want to mislead you.
आपने आज खुद बताया है आपने लेक्चर में कि 65% of water absorption is in the small intestine.
Yes.
Okay.
So it is the increase water content in the colon.
Right. That is true. ठीक है जी. चूंकि अक्सर गलती ये होती है solidification of the fecal material से mislead हो जाते हैं हम कि major absorption occurs in the large gut, whereas 65% occurs in the small intestine.
Okay, so answer is fine. ठीक है.
Yeah.
सर एक क्वेश्चन ये पूछना था कि मतलब प्रोटीन मेटाबॉलिज्म और डाइजेशन की मतलब सारी चीजें हम लोग के उसमें गट में मौजूद है, तो फिर इन चिल्ड्रन बिफोर एज ऑफ 6 months इम्यूनोग्लोबुलिन्स कैसे डायरेक्टली एब्जॉर्ब हो जाती हैं फ्रॉम मदर्स मिल्क?
देखिए मैं आपको बताऊं, when the baby is born, all the tissues and cells of the body, they are in the many of them they have been formed, but some of them they continue to develop up till even 2.5 to 5 years of age. ठीक, many of the components, ठीक है, अक्सर homeostatic regulatory processes they are continue to develop. अभी 5 साल में वो कंप्लीट हो जाते हैं. इसीलिए जो हमारा जो इन्फेंट मोर्टेलिटी रेट ज्यादा है हम जैसे मुल्कों में, उसकी मेन वजह यही है because the children are not taken care, and that some tissues and organs of the body and the systems, they are still in the process of evolution towards completion, which continues even up till 2.5 to 5 years of age. Like, so इस वजह से क्योंकि उनमें जो physiology altogether is different because many of the cells and tissues, they are in the stage of development. That is why. इसी वजह से अगर उनको बुखार होगा तो 106 तक चला जाएगा. हमें बुखार होगा अगर 101 होगा तो हम कहेंगे हमें बहुत बड़ा बुखार आ गया. एंड इफ यू गिव देम एंटीपायरेटिक, थोड़ी सी टिक करके 98 या 96 तक डाउन हो जाएगा, बिकॉज देयर रेगुलेटरी मैकेनिज्म, processes, tissues, cells, and secretions are still in the process of evolution, development, which are completed by the age of 5 years.
तो सर फिर जो मदर्स मिल्क में प्रोटीन्स मिल रही है वो भी मतलब प्रॉपर्ली एब्जॉर्ब नहीं होती या मेटाबोलाइज नहीं होती?
नहीं. अब प्रोटीन इसमें है, which protein can be absorbed quickly and then which proteins cannot be absorbed quickly. जो naturally occurring protein हैं, जो मिल्क में है, मदर्स मिल्क में है, वो हो जाएंगे. जो naturally occurring मिल्क नहीं है, say for ingredients from the other species or ingredients which have been synthesized in the mill or a factory, they will not be able, some of them may not be able to be absorbed because some of the proteins, they possess the property of species specific. जैसे हार्मोन्स में आपने देखा कि कुछ हार्मोन्स के पहले H लिखा होता है, स्मॉल h, ह्यूमन कोरियानिक गोनाडोट्रोफिन, ह्यूमन ग्रोथ हार्मोन. इंसुलिन के साथ नहीं है H. So this is the property of species specific, that the deficiency of a substance or a protein or a hormone of the human cannot be overcome by administration of the same hormone or substance isolated from the other species. So there are many factors, various types of protein and naturally occurring protein, ठीक है जी, तो जिनमें कुछ की होगी, कुछ की नहीं होगी, but it will complete its development by the age of 5 years.
Okay, thank you.
Okay, sir, one last question. What is the
जी.
time taken by food to reach large intestine from pylorus?
हाँ, वो एक to the, मैं जी आपको चलो, अब मैं इसको आपको एक स्लाइड दिखाता हूँ in response to your, वो मैंने जान के काफी सारी स्लाइड्स मैंने, सॉरी ये मूव नहीं हो रहा, काफी सारी स्लाइड मैंने नहीं बताई थी आपको. Let's go to the slide which gives answer to your question precisely. Yes. Look at this slide. The food substance coming from the oral cavity and the esophagus, I'm talking of the mixed food, not water, not fat content, totally mixed food, normally balanced meal. Now its passage, like within the esophagus, is hardly 10 seconds from the oral cavity to reach the stomach. It stays in the stomach for 3 hours. it stays in the small intestine for 7 to 9 hours. Like it stays in the colon by about 25 to 30 hours. That is why generally with a mixed balanced diet, we defecate once in 24 hours because it stays there from taking the food up till 25 to 30 hours. And then what is constipation? If we don't pass fecal material, like up to 120 hours, 30 to 120 hours. If it is, we don't pass fecal material beyond 120 hours, then this will be called as constipation, right? So it stays in the small intestine 7 to 9 hours from the intake of food. And it stays in the large intestine, time taken from intake of food up till 25 to 30 hours, right? Any other question, please?
सर बच्चे में भी यही होगा?
नहीं जी, ये तो मैं adult की बात करूं after 5 years. 5 years onwards, yes. 5 years onwards, yes. But depends on the food. I'm talking of the balanced food taken, ये जो बुलाई जाती हैं slates, balanced food in normal quantity taken at time. बच्चे बादशाह हैं मर्जी के जी, हर वक्त जो मर्जी है खाते रहें, जो गंद बुला खाते रहें, so this will change. This will confuse the intestine and the timings would be changed, maybe change. Yes, please. Right. If there are no questions, thank you very much indeed. It has been a pleasure. We have gone through introduction to endocrinology and introduction to the major functions of the gut. Inshallah, you have been a very recipient because online teaching and learning is boring and a bit difficult because you don't have face-to-face contact. So you can't read the readings in your mind, on the face, on your lips, by your sideways, and by your body language. But it has been your pleasure, and I wish you all the best of luck in your forthcoming exam. God bless you all, and my wishes will accompany you in the examination. God bless you all. Thank you very much. Thank you.
Thank you, sir.
Thank you, sir.
Thank you so much, sir.
You are welcome.
Thank you, sir.
God bless you. Oh, thank you. God bless you all.
Thank you.
CPSP Karachi, can you hear me, please?
Yes, I can hear you.
You
MashaAllah.
can
What
hear.
good
Thank you very
lecture.
much, Eve. So Alhamdulillah, my assignment is over, and see you, inshallah, sometime, somewhere, if not, then in the next course, inshallah, with more chapters and more contents, inshallah.
In the next course, inshallah, probably in March and April.
Thank you very much. God bless you. Thank you.
Thank you so much. Thank you.
Allah Hafiz.
Allah Hafiz. Allah Hafiz. Jazakallah, Jazakallah.
Jazakallah. Ameen.
Participants will take only 10 minutes break because of Jummah prayers. So we'll come back at 11:10 or 11:15 in between. Okay? You all are ready? It's okay for you people?
Yes, ma'am. Yes, ma'am.
So, we'll come back at 11:10. Okay? Dr. Farzan will be there. ताकि हम कोशिश करें कि नमाज prayers पे खत्म कर दें. Okay?
Ma'am, excuse me.
Listen.
If everybody is ready, तो हम चूंकि एग्जाम करीब आ गए, तो if हम रोजाना 10 minutes की break लें